A warm good morning, dear students. Welcome back to biological databases. We have come across different databases in the previous classes of bioinformatics, and let's explore today some of the few databases which are remaining unexplored. And hope you remember the GenBank as well as the Protein Data Bank, as well as various other data banks which we utilized to find out the 3D structure as well as the interactions between various proteins. So let's explore today various other databases which are still remaining. So let's continue with the databases. Previously, we had mentioned about the SCOP or the Structural Classification of Proteins database. It's a database where we had classified the proteins based upon their structural domains, especially based upon the similarity between their structures and the amino acid sequences. But there is a similar database also which classifies proteins, and today we're going to explore them. That is the CAT, the KCAT Protein Structure Classification Database, which is a free, publicly available online resource which provides information on the evolutionary relationships of the protein domains. This was created in the mid 1990s by Professor Christian Orengo and his colleagues, including Janet Thornton and David Jonas, and it continues to be developed by the Orengo Group at the University College of London. This database mainly shares broad features with the SCOP resource. However, there are also many areas in which the detailed classification is being found, and it differs from the SCOP classification also in some aspects. So what you have to remember in your mind is basically you're coming across two protein classification databases. One is the CATH as well as the SCOP. Do remember them, and do explore the database and provide a report on what is the various features of CATH. This is an assignment which I thought you can do rather than I taking you into every database, each and every database as we did the previously. Now, in next database that we have is a VIDA. VIDA is a new virus database that organizes open reading frames from partial and complete genome sequences from the animal viruses. Now you have different animal viruses which are there, and all these viruses would be having various genomic sequences, as well as there would be in each of them there will be what happens open reading frames. So the open reading frames of all these animal viruses are being stored in the VIDA sequence. Okay, and VIDA, it includes the sequences from the GenBank from herpes viridae, corona viridae, as well as the artery viridae. And the open reading frames are organized into homologous protein families, which are identified on the basis of the sequence similarity relationship. And the conserved sequences, the regions of the potential functional importance are identified, and they are retrieved as a sequence alignment. And you can see that a controlled taxonomical and the functional classification for all the proteins and the protein families is being conducted in the database. These, you should remember, it mainly concerns with the open reading frames of the viruses. And when protein sequences, all the classifications which are being classification of proteins which are being coded by these is also being done, and when available, the protein structures that are related to the families have also been included. And the database is online search, and the sequence information retrieval is also there. You can just explore this site. We'll just have a short look soon. So now we'll just explore the VIDA database. I gave the virus database. If you give VIDA, there are lots of things which come, but there is the virus database. That's what I clicked onto it. But I guess this database is having some trouble, but because they are saying that it is temporarily unavailable. So basically, just remember, it's a database which is saving the homologous protein families derived from open reading frames of the complete as well as the partial virus genomes. Now let's explore the KEGG database. KEGG is a database resource for understanding high-level functions and utilities of the biological system, such as the cell, the organism, as well as the ecosystem. And from the molecular level information, we do also reach into a large scale of molecular data generated by genome sequencing or other throughput experimental technologies. Now, when I tell this like this, you might find it, oh, what is it? It has been found to be highly complicated. Let's explore the website so then that will be more easier for you. KEGG is in other words, it is referred to as a Kyoto Encyclopedia of Genes and the Genomes. And in this database, you can see that there are various other sub-databases. You have got KEGG Pathway, KEGG Bright, KEGG Module, KEGG Authology, KEGG Genome, Genes, KEGG Drug, KEGG Disease, KEGG Enzyme, KEGG Reaction, and all that. So that is the reason why I told you it's a database which will explain high level functions. That is from the level of a genome to the level of a protein. Then what is the protein's role in different databases like that, and what are the different drugs which can be used? What are the enzyme classifications? These proteins in what all biochemical reactions they are being exploring. Like that, you have different databases linked to the Kyoto Encyclopedia of the Genes and the Genomes. So basically, it's an encyclopedia, an online encyclopedia, which gives us links to genomes, proteins, as well as what are the roles of these proteins in various pathways, and what are the compounds which are being present. Now, I'll just explore one for your understanding. One is a KEGG pathway. In the KEGG pathway, in what all pathways which are there? Like if you go to have in drug development, what all pathways are there? In metabolism, what all pathways are there? These are all being identified in the KEGG pathway database. Now let's go into KEGG drug. Okay, it gives us information on the approved drugs in Japan, USA, and Europe. You can search their names. Now, if you have a particular component and you want to check whether it is approved, then at the same time, the drug classifications, what are the targets, these are all being explored. So KEGG is an encyclopedia of, it's a Kyoto Encyclopedia of Genes and the Genomes. Hope you can explore them as you get time. In addition to these specialized databases, we also do have some what does it means databases which will give restricted information, or which will be restricted down to one particular organism and all that. And one is the Saccharomyces genome database, which gives all the information on the yeast Saccharomyces. And you can have Species 2000. It's an autonomous federation of which gives taxonomies throughout the world. And now let's discuss about the pitfalls. There could be errors in some sequence databases, and there would also be redundancy in the primary sequence databases, and also sometimes false or incomplete gene annotations could also come in a pitfall as a pitfall of the biological database. But the importance of the database it is that it is a storehouse of information, and you can store as well as organize data so that it can be easily retrieved using various search criteria. And also it allows knowledge discovery, because based upon the previous knowledge itself, you can find out further new information. And the secondary databases, they have become the molecular biologist reference library, and it also helps to solve cases where many users want to access the same entries of the data. And also you can also index the data and also help to remove the redundancy of the data in some cases also. Hope so the databases are clear to you now. With this, we come to an end. Thank you.